In this section, we will talk about reabsorption in detail, such as mechanism of reabsorption, routes of reabsorption, sites of reabsorption, regulation of tubular reabsorption, and reabsorption of important substances. Now, firstly, we will discuss the method of collection of tubular fluid. There are two methods to collect the tubular fluid for analysis. First is the micropuncture technique. A micropipette is inserted into the Bowman capsule, and different parts of tubular portion in the nephrons of experimental animals. To collect the fluid samples are analyzed and compared with each other to assess the changes in different parts of nephron. Second method is the stop flow method. In this method, ureter is obstructed so that the back pressure rises and stops the glomerular filtration. That's why this method is called a stop flow method. The obstruction is continued for 8 minutes. It causes some changes in the fluid present in different parts of the tubular portion. Later, the obstruction is released, and about 30 samples of 0.5 milliliter of urine are collected separately at regular intervals of 30 seconds. The first sample contains the fluid from collecting duct. Successive samples contain the fluid from distal convoluted tubule, loops of Henle, and proximal convoluted tubule respectively. And then all the samples are analyzed. This was all about methods of collection of tubular fluid, which are micropuncture technique and stop flow method. Moving towards the mechanism of reabsorption. Basic transport mechanisms involved in tubular reabsorption are of two types. First is the active reabsorption, while second is the passive reabsorption. Active reabsorption is the movement of molecules against the electrochemical gradient, or, in other words, uphill movement. For example, you can see the man moving the stone uphill. It needs liberation of energy, which is derived from adenosine triphosphate, or ATP. Substances reabsorbed actively from the renal tubule are sodium, calcium, potassium, phosphates, sulfates, bicarbonates, glucose, amino acids, ascorbic acid, uric acid, and ketone bodies. Let's move into more detail of active transport. Active transport involves proteins called as transporters that translocate substances across the cell membrane. Primary active transport uses adenosine triphosphate, or ATP, directly. For example, the sodium-potassium ATPase, or the sodium pump, through which 2 potassium moves into the cell and 3 sodium moves out of the cell. Second example of primary active reabsorption is the calcium ATPase. It causes the movement of calcium out of the cell. Third example of primary active reabsorption is the proton ATPase. It causes movement of H+, or proton, out of the cell. Other type of active transport is the secondary active transport. It uses the concentration gradient created by primary active transport as an energy source. This is most commonly the sodium gradient created by the sodium pump, and the latter therefore plays a critical role in renal reabsorption and secretion. Symporters, or co-transporters, transport substances in the same direction. For example, sodium and glucose moving in same direction through co-transporter or symporter. So, symporter is an integral membrane protein that is involved in the transport of 2 different molecules across the cell membrane in the same direction. The symporter works in the plasma membrane, and molecules are transported across the cell membrane at the same time. and is therefore a type of cotransporter, whereas antiporters transport in the opposite direction. Means antiporter is a membrane protein that transports two molecules at the same time in the opposite direction. For example, the proton and sodium moving in opposite direction through antiporter. Later in this video, you will come to know that where these symporters and antiporters are present, and how do they work. So, don't miss it. Second type of reabsorption is the passive reabsorption. Passive reabsorption is the movement of molecules along the electrochemical gradient or downhill movement. As you can see, a man throwing stone down the hill. So this process does not need energy. Substances reabsorbed passively are chloride, urea, and water. Now, let's discuss the route of reabsorption. Reabsorption of substances from tubular lumen into the peritubular capillary occurs by two routes. First is the transcellular route. Second is the paracellular route. This is the tubular lumen. Tubular lumen is the inside space of a tubular structure. This is the tubular cell, while this is the capillary. Tubular epithelial cells are connected with their neighboring cells by tight junctions at their apical luminal edges. But beyond the tight junction, a small space is left between the adjoining cells along their lateral borders. This space is called as lateral intercellular space. The interstitium extends into this space. First is the transcellular route. In this route, the substances move through the cell. It includes transport of substances from the tubular lumen into tubular cell through apical or luminal surface of the cell membrane. Then from tubular cell into interstitial fluid, and then from interstitial fluid into the capillary. Second is the paracellular route. In this route, the substances move through the intercellular space. It includes the transport of substances from tubular lumen into interstitial fluid present in lateral intercellular space through the tight junction between the cells, and from interstitial fluid into capillary. Sometimes, the substances can also change its route from transcellular to paracellular. Now let's discuss the sites of reabsorption. Reabsorption of the substances occurs in almost all the segments of tubular portion of nephron. First is the substances reabsorbed from proximal convoluted tubule. About 7/8 part of the filtrate, which is about 88%, is reabsorbed in proximal convoluted tubule. The brush border of epithelial cells in proximal convoluted tubule increases the surface area and facilitates the reabsorption. Substances reabsorbed from proximal convoluted tubule are glucose, amino acids, sodium, potassium, calcium, bicarbonates, chlorides, phosphates, urea, uric acid, and water. Second is the substances reabsorbed from loop of Henle. Substances reabsorbed from loop of Henle are sodium and chloride. Third is the substances reabsorbed from distal convoluted tubule. Sodium, calcium, bicarbonate, and water are reabsorbed from distal convoluted tubule. Now, moving towards the regulation of tubular reabsorption. Tubular reabsorption is regulated by three factors. First is the glomerulotubular balance. Second is the hormonal factors, while third is the nervous factors. Let's discuss the glomerulotubular balance. Glomerulotubular balance is the balance between the filtration and reabsorption of solutes and water in kidney. When glomerular filtration rate increases, the tubular load of solutes and water in the proximal convoluted tubule is increased. It is followed by increase in the reabsorption of solutes and water. This process helps in the constant reabsorption of solute, particularly sodium, and water from renal tubule. Moving into further details to understand the mechanism of glomerulotubular balance. Glomerulotubular balance occurs because of osmotic pressure in the peritubular capillaries. When glomerular filtration rate increases, more amount of plasma proteins accumulate in the glomerulus. Consequently, the osmotic pressure increases in the blood by the time it reaches efferent arteriole and peritubular capillaries. Due to plasma proteins such as albumin increase. The elevated osmotic pressure in the peritubular capillaries increases reabsorption of sodium and water from the tubule into the capillary blood. Now let's discuss the hormonal factors. So, the hormones that regulate glomerular filtration rate are aldosterone. Aldosterone increases sodium reabsorption in ascending limb, distal convoluted tubule, and collecting duct. Second hormone is the angiotensin II. Angiotensin II increases sodium reabsorption in proximal convoluted tubule, thick ascending limb, distal convoluted tubule, and collecting duct, but mainly in proximal convoluted tubule. Third is the antidiuretic hormone. Antidiuretic hormone increases water reabsorption in distal convoluted tubule and collecting duct. Fourth is the atrial natriuretic factor. Atrial natriuretic factor decreases sodium reabsorption. Fifth is the brain natriuretic factor. Brain natriuretic factor decreases sodium reabsorption. Sixth is the parathormone. Parathormone increases reabsorption of calcium, magnesium, and hydrogen, but decreases phosphate reabsorption. And seventh is the calcitonin. Calcitonin decreases calcium reabsorption. Third is the nervous factor. Activation of sympathetic nervous system increases the tubular reabsorption, particularly of sodium, from renal tubules. It also increases the tubular reabsorption indirectly by stimulating secretion of renin from juxtaglomerular cells. Renin causes formation of angiotensin II, which increases the sodium reabsorption. Now, let's discuss the threshold substances. Depending upon the degree of reabsorption, various substances are classified into three categories: high threshold substances, low threshold substances, and the non-threshold substances. First is the high threshold substances. High threshold substances are those substances which do not appear in urine under normal conditions. The food substances like glucose, amino acids, acetoacetate ions, and vitamins are completely reabsorbed from renal tubules and do not appear in urine under normal conditions. These substances can appear in urine only if their concentration in plasma is abnormally high, or in renal diseases when reabsorption is affected. So these substances are called high threshold substances. Next is the low threshold substances. Low threshold substances are the substances which appear in urine even under normal conditions. The substances such as urea, uric acid, and phosphate are reabsorbed to a little extent. So these substances appear in urine even under normal conditions. Third is the non-threshold substances. Non-threshold substances are those substances which are not at all reabsorbed and are excreted in urine irrespective of their plasma level. The metabolic end products, such as creatinine, are the non-threshold substances. Now, let's discuss the transport maximum or TM value. Tubular transport maximum, or TM, is the rate at which the maximum amount of a substance is reabsorbed from the renal tubule. So, for every actively reabsorbed substance, there is a maximum rate at which it could be reabsorbed. For example, the transport maximum for glucose, or TM value of glucose, is 375 mg per minute in adult males, and about 300 mg per minute in adult females. Now, let's discuss the threshold level in plasma for substances having TM value. Renal threshold is the plasma concentration at which a substance appears in urine. Every substance having TM value has also a threshold level in plasma or blood. Below that threshold level, the substance is completely reabsorbed and does not appear in urine. When the concentration of that substance reaches the threshold, the excess amount is not reabsorbed, and so it appears in urine. This level is called the renal threshold of that substance. For example, the renal threshold for glucose is 180 mg per deciliter. That is, glucose is completely reabsorbed from tubular fluid if its concentration in blood is below 180 mg per deciliter. So the glucose does not appear in urine. When the blood level of glucose reaches 180 mg per deciliter, it is not reabsorbed completely. Hence, it appears in urine. Now, let's discuss the reabsorption of important substances, majorly in proximal convoluted tubules. First is the reabsorption of sodium. From the glomerular filtrate, 99% of sodium is reabsorbed. 2/3 of sodium is reabsorbed in proximal convoluted tubule, and remaining 1/3 in other segments except descending limb and collecting duct. Sodium reabsorption occurs in 3 steps. Let's discuss the reabsorption of sodium. This is the tubular lumen. Tubular lumen is the inside space of a tubular structure. This is the tubular cell, while this is the capillary. This is interstitial fluid. And tubular epithelial cells are connected with their neighboring cells by tight junctions at their apical luminal edges. But beyond the tight junction, a small space is left between the adjoining cells along their lateral borders. This space is called lateral intercellular space. First step is the transport from lumen of renal tubules into the tubular epithelial cells. Second step is the transport from tubular cells into the interstitial fluid, while third step is the transport from interstitial fluid to the blood. Now let's discuss these steps in detail. Transport from lumen of renal tubules into the tubular epithelial cells means active reabsorption of sodium ions from lumen into the tubular cells occurs by 2 ways. First way is by antiport, which is sodium counterport protein, in proximal convoluted tubules, through which exchange of hydrogen by sodium occurs. An antiporter is a membrane protein that transports 2 molecules at the same time in the opposite direction, while other way is by symport, which is sodium cotransport protein in other segments and collecting duct. Symporter is an integral membrane protein that is involved in the transport of 2 different molecules across the cell membrane in the same direction. The symporter works in the plasma membrane, and molecules are transported across the cell membrane at the same time and is therefore a type of cotransporter, through which movement of sodium, along with other substances like glucose and amino acids occurs. Next is the transport from tubular cells into the interstitial fluid. Sodium is pumped outside the cells by sodium-potassium pump. This pump moves 3 sodium ions from the cell into interstitium, and 2 potassium ions from interstitium into the cell. tubular epithelial cells are connected with their neighboring cells by tight junctions at their apical luminal edges. But beyond the tight junction, a small space is left between the adjoining cells along their lateral borders. This space is called lateral intercellular space. The interstitium extends into this space. Most of the sodium ions are pumped into the lateral intercellular space by sodium-potassium pump. The rest of the sodium ions are pumped into the interstitium by the sodium-potassium pump situated at the basal part of the cell membrane. Transport of sodium out of the tubular cell by sodium-potassium pump decreases the sodium concentration within the cell. This develops an electrochemical gradient between the lumen and tubular cell, resulting in diffusion of sodium into the cell. Now, how transport of sodium from interstitial fluid to the blood occurs? Well, from the interstitial fluid, sodium ions enter the peritubular capillaries by concentration gradient. A concentration gradient occurs when the concentration of particles is higher in one area than another. In passive transport, particles will diffuse down a concentration gradient from areas of higher concentration to areas of lower concentration until they are evenly spaced. A common example of this is a cup of water that you drop food coloring into. The food coloring is concentrated when dropped into the cup of water. However, after a few seconds pass, the particles become lighter in color as they move to the lower concentration level. In the distal convoluted tubule, the sodium reabsorption is stimulated by the hormone aldosterone secreted by adrenal cortex. Now let's discuss the reabsorption of water. So the reabsorption of water occurs from proximal and distal convoluted tubules and in collecting duct. Reabsorption of water from proximal convoluted tubule, called as obligatory water reabsorption. Obligatory reabsorption is the type of water reabsorption in proximal convoluted tubule, which is secondary or obligatory to sodium reabsorption. When sodium is reabsorbed from the tubule, the osmotic pressure decreases. It causes osmosis of water from renal tubule, reabsorption of water from distal convoluted tubule, and collecting duct, called as facultative water reabsorption. Facultative reabsorption is the type of water reabsorption in distal convoluted tubule and collecting duct that occurs by the activity of antidiuretic hormone. Normally, the distal convoluted tubule and the collecting duct are not permeable to water. But in the presence of antidiuretic hormone, these segments become permeable to water, so it is reabsorbed. Now, let's discuss the mechanism of action of antidiuretic hormone through equiporins. Antidiuretic hormone increases water reabsorption in distal convoluted tubules and collecting ducts by stimulating the water channels called aquaporins. Antidiuretic hormone combines with vasopressin receptors in the tubular epithelial membrane and activates adenyl cyclase to form cyclic AMP. This cyclic AMP activates the aquaporins, which increase the water reabsorption. Aquaporins are the membrane proteins which function as water channels. Though about 10 aquaporins are identified in mammals, only 5 are found in humans. Aquaporin 1, 2, and 3 are present in renal tubules. Aquaporin 4 is present in brain, and aquaporin 5 is found in salivary glands. Aquaporin 2 forms the water channels in renal tubules. Let's make a comparison of responses to water intake and responses to water deprivation. In case of responses to water deprivation, increases plasma osmolarity, stimulates osmoreceptors in anterior hypothalamus, increases secretion of antidiuretic hormone from posterior pituitary, increases water permeability of late distal tubule and collecting duct, increases water reabsorption, increases urine osmolarity, and decreases urine volume. Decreases plasma osmolarity toward normal, but in case of responses to water intake. Decreases plasma osmolarity. Inhibits osmoreceptors in anterior hypothalamus. Decreases secretion of antidiuretic hormone from posterior pituitary. Decreases water permeability of late distal tubule and collecting duct. Decreases water reabsorption. Decreases urine osmolarity and increases urine volume. Increases plasma osmolarity toward normal. Moving towards the reabsorption of glucose. Glucose is completely reabsorbed in the proximal convoluted tubule. As we have discussed that, this is lumen. These are tubular cell. This is interstitial fluid. And this is the capillary. Glucose moves from tubular lumen to tubular cells. Then from tubular cell to interstitial fluid. The transport of glucose from tubular lumen to tubular cells is by secondary active transport or sodium cotransport mechanism. Glucose and sodium bind to a common carrier protein in the luminal membrane of tubular epithelium and enter the cell. The carrier protein is called sodium-dependent glucose cotransporter 2. From tubular cell, glucose is transported into medullary interstitium by another carrier protein called as glucose transporter 2 or GLUT2. In adult male, tubular maximum for glucose is 375 mg per minute, and in adult females, it is about 300 mg per minute. Renal threshold for glucose is 180 mg per deciliter in venous blood. When the blood level reaches 180 mg per deciliter, glucose is not reabsorbed completely and appears in urine. Now, let's plot a graph and understand few terms. This is x-axis, showing glucose reabsorption, and its unit is milligram per minute. This is y-axis, showing plasma glucose, and its unit is milligram per deciliter. Now, what is splay? Splay means deviation. With normal glomerular filtration rate of 125 mg per minute and tubular maximum for glucose of 375 mg per minute in an adult male, the predicted or expected renal threshold for glucose should be 300 mg per deciliter. But actually, it is only 180 mg per deciliter. When the renal threshold curves are drawn by using these values, the actual curve deviates from the should be, or predicted, or ideal curve. This type of deviation is called splay. Splay is because of the fact that all the nephrons do not have the same filtering and reabsorbing capacities. Let's move towards the calculation of reabsorption and secretion rate. The reabsorption, or secretion rate, is the difference between the amount filtered across the glomerular capillaries and the amount excreted in urine. Filtered load is equal to glomerular filtration rate into plasma. Excretion rate is equal to volume into urine. So reabsorption rate is equals to filtered load minus excretion rate, and secretion rate equals to excretion rate minus filtered load. If the filtered load is greater than the excretion rate, then net reabsorption of the substance has occurred. If the filtered load is less than the excretion rate, then net secretion of the substance has occurred. Now, let me give you a clinical case. A woman with untreated diabetes mellitus has a glomerular filtration rate of 120 mg per minute, a plasma glucose concentration of 400 mg per deciliter, a urine glucose concentration of 2500 mg per deciliter, and a urine flow rate of 4 mg per minute. Now, what is the reabsorption rate of glucose? As we have discussed that filtered load equals to glomerular filtration rate into plasma glucose concentration. So putting the values 120 milliliter per minute into 400 milligram per deciliter. In next step, 120 milliliter per minute into 400 milligram divided by 100 milliliter. Because 1 deciliter equals to 100 milliliter. These two units are same, so they cancel each other. These zeros also cancel each other. So 4 into 120 equals to 480 milligram per minute. Now, excretion is equal to urine flow rate into urine glucose concentration. Putting the values into the equation. Urine flow rate is 4 milliliter per minute, and urine glucose concentration is 2500 milligram per deciliters. Similarly, 4 milliliter per min into 2500 milligram divided by 100 milliliter, as 1 deciliter equals to 100 milliliters. These signs are same, so they cancel each other. These two zeros also cancel each other. Now 4 into 25 is 100. So the answer is 100 milligram per minute. Final equation is reabsorption equals to filtered load minus excretion rate. It has already been discussed. So putting the values 480 milligram per minute minus 100 milligram per minute. So, reabsorption is equals to 380 milligram per minute. Let's discuss the reabsorption of amino acids. Amino acids are also reabsorbed completely in proximal convoluted tubule. Amino acids are reabsorbed actively by the secondary active transport mechanism by symporters along with sodium. Last is the reabsorption of bicarbonates. All the filtered bicarbonate in the renal tubules is reabsorbed. About 80% of it is reabsorbed in proximal convoluted tubule, 15% in Henle loop, and 5% in distal convoluted tubule and collecting duct. The reabsorption of bicarbonate utilizes the proton secreted into the renal tubules. Bicarbonate is reabsorbed actively, mostly in proximal tubule. It is reabsorbed in the form of carbon dioxide. Bicarbonate is mostly present as sodium bicarbonate in the filtrate. Sodium bicarbonate dissociates into sodium and bicarbonate ions in the tubular lumen. Sodium diffuses into tubular cell in exchange of hydrogen through sodium-hydrogen antiport pump. Bicarbonate combines with hydrogen to form carbonic acid. Carbonic acid dissociates into carbon dioxide and water in the presence of carbonic anhydrase. Carbon dioxide and water enter the tubular cell. In the tubular cells, carbon dioxide combines with water to form carbonic acid. It immediately dissociates into hydrogen and bicarbonate. Bicarbonate from the tubular cell enters the interstitium. There it combines with sodium to form sodium bicarbonate. This was all about this section. In our next section, Dr. Jessica will join us and help us to understand the tubular secretion in detail.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. only on sqadia.com.